In this video, I'm going to go through the enrichment tab of the platform. So first of all, we have the genset enrichment module, which is this one. Here on the left hand side, you have several scroll down menus. The first one allows you to choose a contrast you want to look at. The next one allows you to choose which genset collection you want to look at. By default, it picks them all, but if you specifically just want to look at one, say GO and KEGG pathways, then you can select it from here. You can also change the stringency of your FDR and log4 change thresholds if you want. And under the options, you can click this box if you want to see all gene sets and not just the significant ones. And you can also choose if you want to remove any of the statistical methods, or if you are happy to keep stringent intersection of all three of them. So what do we have here? First of all, here on top, we have under the top enriched tab, we have several genset enrichment plots. They basically show you for this particular contrast what gene sets from public databases are positively, or in this case, you can see they are mostly negatively correlated to them. If we just go down here to the table, we can see a list of all of the gene sets from these public databases that are statistically correlated. So let's order this now. I'm going to order this by MetaQ values. There you go. So now you can see that some of them are positively correlated, some of them are negatively correlated. Also, if you click on one of these gene sets, you can see the list of the genes that compose them, as well as some statistic related to them. Here on the left hand side, you also have a gene frequency plot. Basically, this shows you the number of times a gene that is present in the topmost correlated gene sets is present. You can actually customize this, so by default it shows that the 25 top most correlated gene sets. You can change it to say 100 or 10, and there are other options here that you can change. Let's go quickly back here to the fold change all tab. This is basically just showing you again for the gene sets that are statistically correlated to these particular comparisons, how the enrichment score looks like across all the pairwise comparisons. And for the FDR tables, you have for all the methods that are available and all the comparisons, the number of gene sets that show up as you change the FDR values. So for example, if you put a very permissive FDR values, you have a high number of gene sets showing up, and this goes down to 0 as you make it very, very stringent. If we go now here to the plots tab, we can see first of all a volcano plot that shows you, so let's go back to table. So if you select, for example, a specific gene set, you will see its related volcano plot here with all the genes that compose it highlighted. You can also see an enrichment bar plot that shows you the two groups that we are comparing, and showing you in this case how the genes in this particular gene sets are behaving. And here on the other hand, you can see a specific gene from the same gene set. So for example, here it's showing GPX1s. We can change it to the second one, CD79B, and then you see a different plot. And here we have an enrichment versus expression plot. If we go to the compare tab, now again this is related to the gene sets that you pick here, and you can basically have an overview of how this particular gene set is enriched across all the pairwise comparisons. So you can quickly see which ones are behaving similarly and which ones are behaving differently. The volcano tab shows you again, for all the contrasts, the volcano plots of that are produced by the genset enrichment methods. And if you want on your end to see how different methods compare with each other, you can just go to the Volcano Methods tab, and this is again just looking at the contrast that you selected here, and so you can see how the different statistical methods perform. Finally, under the GenMap tab, you can see a co-activation heat map. Again, this is based on which gene set you select from the table below, and you can see the various gene sets that are correlated with it, and the genes, the topmost represented genes across these gene sets. So let's move on to the next tab, which is pathway analysis. This is pretty obvious. Again, on the left hand side we have a scroll down menu where you can choose your various contrasts. You have some options here which you can activate if you wish. And basically what this does is some standard KEGG pathway and GO terms analysis. So for the KEGG pathway you have a nice visual depiction of the KEGG pathway. We can enlarge it here to see more in detail the various genes and how they are up or down regulated. Then we have then we have a list of the various KEGG pathways that are present that are present, and you can sort them by log fold change if you want, or by meta Q values. And you also have an activation matrix that basically shows you across all the pairwise comparisons which of these KEGG pathways are significantly up or down regulated here. And the same is pretty much true for the GO term analysis. So you first of all get a gene ontology graph. This is an interactive graph, so you can play around with it if you wish, and you can maximize it to see all the various terms that are significantly identified by the platform. Here you have a table again with all the various GO terms and the statistics related to them, and the activation matrix as before that shows you across all the pairwise comparisons which of the GO terms are significantly up or down regulated. Next we have the word cloud tab. So again, from the word cloud tab you can select a contrast with the left hand side scroll down menu. There is an option here that allows you to normalize the activation matrix. I'm going to talk about that in a second. And before I go on to explain what different plots do, what does this tab do? Well, actually, it takes the results from the genset enrichment analysis and extract the most common words that are found across them. So if we look at the table here, we can see that for this particular contrast, the most common word is rat. And we can then do some enrichment plots for all the available contrasts and see how the word rat is positively correlated for most of them. If we take a different keywords, for example, hematopoietic, we can see that for the first three pairwise comparisons there is a positive correlation, but this becomes a negative or non-significant correlation for the bottom three. The platform also produces a word cloud plot. It basically takes the same keywords from the table and displays them by changing the font size and intensity of the color according to their frequency. You can also change this. For example, here I already excluded the word rat because in this case it didn't make sense. I could reinstate it just by deleting it. So you can see now rat shows up in my word cloud, but I prefer to remove it because it doesn't make sense in this case. and I can also change the color of the word cloud. So for example, I can turn it into shades of gray. There is a word TSNE plot. Actually, this is both a TSNE and UMAP plot. You can change it here from the settings menu. And there is an activation matrix, so the one I just mentioned before with the options here. This is basically the same as what we saw for the KEGG pathway and Go term analysis. It takes, in this case, the keywords and shows how frequent they are present in the various pairwise comparisons. Also, we do have a leading edge table. What it does is basically takes a keyword and sees where this keyword appears in the gene sets that we have found in our analysis. So for example, you can see the word rat is present mostly because most of these samples were probably derived from rat experiments. The last module within the enrichment tab is the drug connectivity module. What the drug connectivity module allows you to do is to take the gene profiles from one of your contrasts and compare them against the gene profiles from drugs obtained from 3 different public databases, namely the L1000, the CTRP version 2, and the GDSC databases. So the platform will first of all give you a list of all the drug profiles that are correlated to your contrast gene expression profile, and you can order this by normalized enrichment scores, for example. So in this case I already ordered them from most positively correlated to least to most negatively correlated profiles, and then it will take the top hits and display them as gene set enrichment plots here. Now, if you're looking at the positively correlated ones, if you're interested in understanding, for example, the mode of action of a drug, then you want to look at the most positively correlated drug profiles. And the plot here actually shows you the frequency of the most common modes of action of the drugs in the list. So for example, for this particular comparison, topoisomerase inhibitors as well as mTOR and MEK inhibitors appear to be the most positively correlated. So this could indicate potential mode of action for your compound. On the other hand, if you were looking at a disease, you might want to look at most negatively correlated modes of action as indicating drugs with potential therapeutic potential. The last thing I want to point out is the activation matrix. This is basically showing a summary of the correlation of the various drugs across all the pairwise comparisons that you have in your dataset.